our cell contains a lot of different types of organelles. If you remember back to the first discussion about this lecture, organelles, it's aptly named. It's well named because it's kind of like this is a little dude or young lady floating around and has the little organs that do the specific different types of functions that only they can do. I believe I've starred this one quite nicely. This is one thing I would highly recommend memorizing, all the bits and pieces and parts. Label them appropriately, know what each part does and how it does it. To how far we've described it here in the book, as well as here in this lecture. So, we've talked the cell membrane, let's talk about the inside. We got the cytosol, the cytoplasm. The cytosol is like your inner liquid matrix of the cell. It's the liquid inside. Intracellular fluid. Within that intracellular fluid is suspended or found inside this intracellular fluid are little organelles that are specialized cells. Here's the list of all the organelles that I would recommend memorizing. Let's go ahead and take a look-see at the different types first, starting off with the skeleton of the cell. This is called the cytoskeleton. Cyto again means cell, skeleton means, well, skeleton, right? You got 3 different types of fibers that make it up. You got microfilaments that are, if you notice, they're really found on the outside perimeter of the cell. You find it in the microfilaments. You find it on the outside boundary. They're there sort of as a structural support, like that crunchy outside of one of those dum-dum suckers. The intermediate filaments is sort of trying to connect and suspend all the different types of organelles and holding its shape, while the microtubules are the networks. They are the streets and roads. There are things inside the cytoplasm that isn't just floating willy-nilly, often and frequently inside the cytoplasm. It's actually being transported. If you're remembering exocytosis, endocytosis, how do they know which way to go? Well, it's because these endotubules have little feetsies that hold, or a little machine that holds on to them, that grabs onto that vesicle and moves it around. You'll also see when we're talking about DNA and cell replication and mitosis that these microtubules also have a huge role in moving our DNA to the appropriate sides of the cell. Now, the main hub for all of these roads that are the microtubules are found at the centrioles. This is also called a centrosome. Centrosome is a singular word for it. It's a permanent structure. Microtubules usually aren't permanent. They can be reorganized, but these right here will not be reorganized. These are centrosomes. They are the center. You do not dissolve the center unless you want some really bad things to occur. Something else that these microtubules can be a part of is the cilia or flagellum. In bacteria, you can see both cilia and flagellum quite often in a single cell organism. But in us and in tissues, since they're stationary, cilia often are there to help move stuff on the outside. When you're coughing, a good majority of the reason why you're able to expel the liquid in your lungs or that mucus in your lungs is because you have little cilia moving that junk up and out. Smokers cough. Why it's so unproductive. You can't really cough up that thick mucousy phlegm is because a lot of the times that smokers' lungs, all that smoke have destroyed these cilia, and that mucus can't be moved out as easily. End of FYI on that one. You can also have a very large structure called the flagellum that looks like a tail, which you can find on the end of a sperm cell. Flagella can move in a whip-like fashion, while the sperm or the flagella of the sperm or whatever cell we're going to be talking about in microbiology will actually be propelling forward in a corkscrew type fashion. that is your cytoskeleton. That is how you actually hold your shape, how certain tissues look the way they are. Let's talk about ribosomes now. Ribosomes are your protein makers. Certain little molecules are brought to this ribosome that says, hey, we need to make this, and the ribosome does great at doing so. You'll find ribosomes everywhere. You'll find it free-floating the cytosol. You'll find it embedded in the endoplasmic reticulum as well. In fact, we can discern the different parts of the endoplasmic reticulum based on the presence of those ribosomes. Again, ribosomes are the protein makers. You'll see a rough endoplasmic reticulum and a smooth endoplasmic reticulum. The rough endoplasmic reticulum is, by the way, an endoplasmic reticulum is a lipid membrane embedded with a whole bunch of different functionalities. We'll say that the rough endoplasmic reticulum, do a quick thought exercise with me, if ribosomes make protein, and these ribosomes are embedded in fats, most likely they're going to be making proteins that interact with the cell membrane, right? Because if you make a non-water loving thing out in the cytosol, it's going to have some problems. So the rough endoplasmic reticulum has a lot to do with the formation and creation of proteins that are going to be expelled outside the body or are going to be part of the cell membrane. The smooth endoplasmic reticulum has a lot to do with making stuff that is fat-loving, but can't really be made in the cytosol, so this is where it goes. The endoplasmic reticulum will then send it off. Here's some pretty pictures of the smooth and rough endoplasmic reticulum. We'll then send it off to the Golgi complex. They've made this say, hey, it's time to move this stuff on. We need this to go. And the Golgi complex is like the postal service. Little vesicles are going to be sent from the rough endoplasmic reticulum or the smooth endoplasmic reticulum, and it's going to be sent to this Golgi apparatus, where it is further processed. You don't need to know how it's processed. Just know that certain things occurred that allow it to be stamped, postmarked, and off it goes, often to the cell membrane to be the contents to either be released out into the wild blue yonder or to become part of the cell membrane. Here's a picture of it occurring. Again, make protein or make something, send it to the Golgi complex. It puts a stamp on it, and it's going to move these vesicles around until finally it gets sent out to the outside, where it either becomes a part of the membrane or it's released out into the wild blue yonder. You may have remembered from the vesicle portion that there's these little lysosomes right there. A lyse means to break down or destroy. So a lysosome, when you hear lysosome, you should think break down and destroy. So when something like a white blood cell comes along, a lysosome is going to, and that white blood cell eats something, a lysosome is going to join onto that little vesicle that's made and break it down and release lysozymes, digestive enzymes. You also have something called a peroxisome. Peroxisomes are pretty similar to what they do, except for they make oxygen. An oxygen, or a hydrogen peroxide, I should say, which makes it, you'd be surprised that oxygen is not our best friend. It's one of the reasons why, more often than not, why our cells degrade quite easily. But I digress. We'll talk about that later. Peroxisomes can also destroy a lot of the organic materials and substances. Finally, one other degradational thing that is inside our cells is called the proteasome. If we take apart that word again, we have protein at the front and zome. We've seen lysosome, peroxisome, anything with the zome at the end of it seems to break down something. So if we say proteasome, we're most likely saying something that breaks down protein. Protein we no longer want or need. With the zomes done, let's move on to the powerhouse. We got ourselves the mitochondria. A lot of you have probably heard that from your high school. Hopefully it's stuck. It'll make your life a little bit easier if you remember it. But the mitochondria is the powerhouse. This is the place that creates so much energy for your cell that if you didn't have a mitochondria, you would be out of luck. You would not be existent right now. Mitochondria, just a quick FYI, you'll probably need to know this later on in life, aka in P4, are passed on only by the mother. So if you ever wanted to find the maternal line, your maternal line, you'd only have to use mitochondria. None of that messy stuff that comes from both mother and father that's in your nucleus, but your mitochondria only has DNA from mother. Finally, the nucleus. Let's talk about the nucleus. The nucleus contains your genetic information. It has all of your DNA housed inside of it, with the nucleolus being the place that you have a lot more of your ribosomal RNA and other things being created in the middle. The nucleus, usually the DNA is pretty loosey-goosey. You can't really see any structures. The only time that you can actually see these little characteristic X's that we see in DNA, or during mitosis, is, well, during mitosis, because when you want to divide something off or duplicate and move it along, you don't really want too many loose ends, or else they're going to break off and you're going to have some issues. You don't want this X structure when you are in normal functioning mode, not duplicating a cell, because if you do, then you're not going to get to your genetic information and you're not going to be able to produce and be a viable cell. Now, there are some terms I should hit on really quick before I move on, and that is that these are chromosomes. One X is called a chromosome. One side of an X is called a chromatid. That's going to be important. A centromere is just the center of that chromosome. With that, that is the end of that portion of our lecture. We'll be discussing protein synthesis in the next one.